from Microbe TV, this is TWiP, This Week in Parasitism, episode 140, recorded on October 16th, 2017. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. Get $30 off your first delivery and free shipping by going to blueapron.com/TWiP. Hi, everybody. I'm Vincent Racaniello, and joining me here in a sunny, lovely New York City, Dickson, Despommier.
Hello, Vincent.
And Daniel Griffin.
Back from the wilds, yes. I am here.
Wow. He's actually not here in New York City.
No, he's not.
He's at a remote location.
That's right.
He is our he's our remote broadcaster.
That's right.
Now, you were away, Daniel? Is that right?
I just got back yesterday from some time in Panama. It was great. A fantastic experience. I was working with an organization called Floating Doctors. Some of our listeners probably have heard of them.
Nice.
And it was an organization started probably about a decade ago. And they initially were in other parts of, I'll say, the Caribbean. But now they've set up shop permanently in Panama, and they are serving the isolated indigenous tribes.
How marvelous.
Wow.
And it was really quite the experience. We left and traveled by about 5 hours by a motorboat to a remote village, and no running water. The water basically it rains and you collect it off the roof. No electricity,
right.
And lots of tropical and parasitic diseases, and just wonderful people.
Is there a dog barking there?
Not on my end. You guys have a dog there at Columbia? I hope
we don't. We do not.
I thought I heard some weird. How long? What did you do there, Daniel, yourself?
They would set up these I guess you call them mobile medical clinics, sort of similar to what I think I described for our listeners when I was up in the remote part of the Dominican Republic Haitian border area. And they had a similar structure. It was a concrete floor. There was a roof. The walls were open. And we would set up an intake where patients would basically be processed. And then the patients would eventually make it to see the clinical groups. And I was one of the clinicians. And they'd present with whatever brought them in, whether it was worms, diarrhea, respiratory infections, skin lesions. Maybe we'll talk about one of those in a later TWiP. And then they had a pharmacy set up, so you could actually set them up with medications. And the group makes a point of visiting each location every 3 months, so that they can then get back and, let's say someone was put on a chronic medicine, and there's some chronic care now really starting to make inroads in the tropics. So hypertension, diabetes.
That's encouraging.
And so, but you got to, if you put someone on hypertension or treat their heart failure, you got to come back. You can't just treat them for 90 days and then walk away. So that's a great thing. This organization, I have to say, was, just like I said about FMRIC, fantastic people, really committed. And I think the thing I appreciate the most is incredibly culturally sensitive. Nice. I mean, the people just greeted us so warmly, not because of me, but because of the relationships that this organization has established over the years.
You said native peoples. Do some of them speak languages other than Spanish or English?
Well, for all of them, Spanish was a second language.
Exactly.
Yeah.
So what was the first language then?
It's a language called Nabay, and that's the tribe, the Nabay tribe. Very interesting social structure. And for them, they were speaking, those that could, Spanish as a second language. And in some cases, there might be a family member who spoke Spanish. That was the interface language, translating for members that did not speak Spanish.
Right.
But there was, I think at the tail end, we spent a little time in a nursing home, interesting enough, right? Nursing home in the tropics and a Asylum. And those mainly women. They live longer than us. The men die off. So there were men, but it was more women than men. And they spoke Spanish as their primary language.
I'm looking here at a webpage. Apparently, there are quite a few languages spoken in Panama. They have 7 different ethnic communities.
That's why I asked.
Wow.
Yeah.
That's very interesting.
Do they speak Papiamento? Is that a language listed there or no?
The dialects are Ngabere. I can't pronounce it. Bolyari, Hakka Chinese, Mbera, and Kuna.
Chinese.
Hakka Chinese.
Hakka Chinese. Wow.
Curious.
Fabulous.
Dickson, you ought to go sometime.
Oh, no, I should. You're absolutely
Have you been?
I haven't. I was very, 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 very close, however.
Very, very, very?
We made a driving mistake.
Very, 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 very?
When we were in Costa Rica,
I'm making fun of someone.
That's okay. We almost drove into Panama and wondered why the traffic suddenly picked up and got very dense. And, of course, that was the border crossing.
Did you see the canal?
You're far away from the canal, that's for sure.
Daniel, did you see the canal?
I saw the canal.
Wow.
Was it impressive?
It really was. I had hoped to arrange a tour of the canal to actually go see it. Unfortunately, there was some very interesting flight challenges on my way back where the original plane was broken, and then they had this idea they would shuttle us in Cessnas all over to another airport where there was a big plane, until one of the fellow passengers came up with this really brilliant idea. If there's a plane at the other airport, wouldn't it be easier to bring the plane to us instead of us 6 at a time to the big plane?
Right.
But by the end of the day, I had missed my window of opportunity to visit the canal.
Too bad.
But I will certainly go back. What a wonderful country, wonderful people.
Great.
Neat. All right, we have one follow-up before we get into our case from last time. And this is from Anthony, who writes, I'd not heard of knockout rats before the TWiP 139 paper discussion. As luck would have it, last Saturday I happened upon a December 2009 issue of Science. On the back cover, there's an ad for knockout rats, and he includes an image, a full-page image from Sigma.
Right.
You can stop daydreaming. Knockout rats are now a reality. And the image is of a cloud with the outline of a rat. Now, Dickson and Daniel, are you daydreaming about knockout rats?
Not often. Not often enough, apparently. No, I don't have to. I can just go to the 59th Street at Columbus Circle subway stop and look down on the tracks, and I can see all the rats I want.
Yes, but this would be in the laboratory.
That's true. I used to work with rats a lot.
Well, anyway, you can buy them, and we know because in that paper they used them.
Yeah.
So there you go. If you dream of it, it can become reality.
No, that's an absolute fact. I totally agree with that. No, I do. If you can imagine something, it can be done. That's right. We will find a way.
Well, Dickson, I can imagine all kinds of zeros, but not going to happen. Okay, let's get back to our case from TWiP 139. Exactly. Daniel, remind us, it's not very long, is it?
No, it hasn't been that long, but a lot has happened since then. So I am glad that we have notes so that I can remember.
Right.
This was a woman in her 30s who had returned to the U.S. after a 2-year period of service in the Peace Corps in Cameroon and Gabon. And we did not know much about her time in service in these areas. But what was of note is when she had her medical exam on return, they had detected Eosinophilia. They had failed to reach any diagnosis at that point. But then she had decided to attend graduate school in New York City at a place where amazing things are happening.
Right. And, nudge, nudge, wink, wink.
And the Eosinophilia is noted again. And
so it didn't go away.
It didn't go away. She's otherwise asymptomatic.
Interesting.
And we left people, I think, there, right, to guess what would you be thinking about in a Peace Corps volunteer from this region of Africa. And we did point out it's in that sort of cradle, almost like an armpit area, sort of almost like a New Jersey of Africa.
Oh, no. Hey, now, wait a minute now. Come on. Vincent and I both live in New Jersey. That's
Oh, I mean it
totally unfair.
No, I mean it just that's a sort of a description of looking at the northern part of Africa juts out, and there's an indentation, almost like an arm reaching out, and I don't mean it.
Try to get out of this. Come on, Daniel, just try.
There's some parts of New Jersey that are quite beautiful.
There are many places in New Jersey
sparsely populated.
That's correct.
Populated. They're nice forests.
Exactly.
Interesting animal life.
That's right. But it's also heavily industrialized, and some people landing in Newark Airport are appalled with the different colored air every now and then, and the smells are not so great.
Improved a lot from when
it has, but still, oh my gosh, everything has. In fact, you can now go swimming in the Hudson River.
Yeah, beautiful beaches in New Jersey, beautiful mountain area.
That's right.
Northwest.
Exactly
you could even eat fish from the Hudson River. I think you're allowed one a week.
Something like that, yeah. But that's not our fault. That's up in the General Electric Corporation.
All right, we have 3 case guesses.
Right.
First is from Wink. Dear TWiP professors, I read that onchocerciasis and loiasis, thank you, can be found in Gabon and Cameroon. I would try to diagnose these in the Peace Corp Veterans case. Onchocerca microfilariae can be found in skin snips from the lower body stained with Giemsa and or by PCR. Loa microfilariae might be found in concentrated midday blood stained with Giemsa or Wright's stains and by PCR if available. I'm thinking that other chronic metazoan infections, such as Bancroftian filariasis, are less common with her history. Thanks as always for the challenging cases and insightful discussions. Wink Weinberg in Atlanta. Now, just explain to me what's with midday blood.
Most of the microfilariae that circulate from filarial parasites, at least, are of 2 varieties. One is periodic, and the other is subperiodic or aperiodic. And in the case of Loa, it's not periodic at all, so it doesn't matter really when you take the blood.
I see.
So you could take it even in the morning or evening, and it would still be there. But in the case of Bancroftian filariasis, Wuchereria bancrofti, then you have to find out whether it's periodic or subperiodic in that region before you decide when to take the blood.
Interesting.
Yeah,
you agree with that, Daniel?
Yeah, I was actually going to say that was one of the reasons why at one point I did not agree to take up a research project in India, because my role was going to be the 2 a.m. blood draw. It was a tropical pulmonary eosinophilia case. Right. So this was going to be one of the Wuchereria.
That's right.
Filaria.
And that is periodic, and it only comes out at night because that's when the mosquitoes that bite come to take it up. So that's
understood.
It's really amazing that the evolution has done that they've optimized to for better transmission. And it does take a little bit of time, and the parasite will change its periodicity when it comes to a new time zone, not right away.
Or with work. If a patient doesn't work during the daytime but works at night, it's completely reversed. No kidding. Because it deals with the reason why the microfilariae accumulate around the lung fields at night, during the daytime rather, is because of the drop in PO2 to O2 ratio, and then the PCO2 rather to O2 ratio. As soon as you go to sleep, that whole thing changes and it releases them from the lung fields, and they go out into the peripheral blood. Then they start to become available for mosquitoes.
Could you take the next one, Dickson?
I would be happy to. Joseph writes, Dear Triple TWiP, for our patient with a travel history to Gabon and Cameroon, the asymptomatic eosinophilia, I would stay more on the conservative side of treatment depending on how severe the eosinophilia is. The most important thing I could do is probably get a more thorough history and a physical exam. Parasites aren't the only thing that cause eosinophilia, so I would ask if there were any new drugs that were started 2 years ago, or a history of allergic symptoms or autoimmune conditions, weight loss or systemic symptoms in these last 2 years could also rarely suggest a malignancy or an allergic eosinophilic syndrome. I would also focus on any free river exposure or food exposures that could suggest a chronic strongyloides, schistosome, or another worm infection. I would get a CBC, complete blood count, and a CMP to monitor if there were any other organs involved, such as iron deficiency anemia due to blood loss or liver enzyme release from schistosomes, and to monitor the eosinophilia. If there are no findings and no other positive lab results, I believe that I would monitor the patient for signs of organ involvement every 6 months to 1 year. I wouldn't be in so much of a rush in order to order more testing without more evidence to suggest a problem. UpToDate states that 50% of people with eosinophilia never have a definitive cause attributed and tend to do fine. I would reassure the patient that is as much as I could do. Sincerely, Joseph Davidoff.
So maybe I would say for our probably a lot of our listeners are not familiar with what UpToDate is. And UpToDate is a resource that a lot of clinicians use. It started a number of years back, and most universities, a lot of medical centers have subscriptions. Individual providers might have subscriptions, and it allows you to look up specific topics.
Got it.
And there are people that are hired. I imagine there's money changing hands, unlike our shows. And,
That's right.
We do it for the love.
We do.
But they allow you to look up and have an updated, as they say, up-to-date review on a topic, whether it's how to treat community-acquired pneumonia or how to approach a patient with eosinophilia, and just to give people what that up-to-date is. What else was there in there that, one thing I wanted to actually commend, so I think what we talked about, what is the cheapest test at its history. It's always good to get more history.
Yeah.
I think I told people from the start you weren't going to get a lot.
That's right.
Cheapest test is history.
It's exactly right.
Yeah,
Just keep talking.
Just keep talking and walking.
That's right. And you have to know when you've heard something, though. That's the other thing. You can listen without actually picking up as to what may be the cause.
You can listen without hearing.
There you go. You can see without seeing.
We have one more, and Daniel, can you take that one?
Lachlan writes, Dearest TWiP, I'm not sure if I'm too late to write in for TWiP 139. I have been on a TWiP binge these past few weeks and can't help myself. A quick note about the Peace Corps woman with eosinophilia. Thinking first about the pace of our evaluation. I did not pick up her absolute eosinophil count from the podcast. However, given that she is asymptomatic and has been in West Africa for 2 years doing good deeds unhindered by her blood count, the problem is probably not urgent. To develop symptoms, of course, there needs to be not only excess eosinophils in the blood, but some pathological factor driving activation and tissue infiltration. Mm-hmm. Not to call you predictable, I suspect infection in this case. In this area, Dr. Griffin has taught us well. My immediate thoughts are of invasive helminthiases such as strongyloides, the odd protozoa out, Cytoisospora belli, Sarcocystis species, and Dientamoeba fragilis, and even possibly intestinal Diphyllobothrium latum infection. Apparently, even ectoparasite infestations like scabies can cause eosinophilia. Mm-hmm. Next, they will be saying that Ekbomb Syndrome can too. I should just throw in, that's I guess a fancy word for delusional parasitosis, or another word that
psychosomatic.
Well, where people, I think, are concerned that they have a parasitic infection, but potentially do not. Common things are common. I suspect strongyloides, as it apparently can cause chronic infection lasting decades, helped by its ability to complete its life cycle in humans, an ability shared only with Capillaria philippinensis, as far as I know. Chronic infection can be asymptomatic in over half of patients and causes intermittent eosinophilia in the majority. Running with this hypothesis, I wonder from whence the worm came. Strongyloides is a geohelminth, and I'm eager to hear whether this woman has a penchant for barefoot activities at home or abroad. Perhaps she went on a shoeless peregrination on a previous trip to tropical parts of Asia.
How
South
dare
America,
she?
or maybe in one of the endemic foci in the United States. Somewhere along the line, she has encountered a third stage filariform larvae, which quickly made its way through the skin, venous system, lungs, and trachea to its new home in the small intestine. Apparently, the parasitic females only live for a few months. Chronicity is explained by autoinfection, whereby eggs hatch into first-stage rhabditiform larvae and are able to complete subsequent molts within the host, after which they penetrate the distal colon or perianal skin to complete their life cycle. The woman has been asymptomatic for years. She probably has a light infection, with her immune system managing to keep the threadworm population at stable, low levels over time. She also may have not taken note of the transient symptoms of larva curans and eosinophilic pneumonia as the worms happily go about their lives. This might sound like a match made in heaven, hardly fitting the bill for parasitism. But diagnosis and treatment now will prevent her from developing hyperinfection in the future as she ages, and should she need to be immunosuppressed for some other medical condition. Diagnosis rests on finding larvae in feces or antibodies in serum. Stool microscopy is insensitive because of intermittent and low-level shedding, requiring multiple specimens over a period of time. Concentrating the specimen or Harari Mori culture might be useful for this. As expected from how quickly the eggs hatch, the usual form in the stool is the first-stage rhabditiform larvae rather than the egg or adult worm. A positive Strongyloides EIA would be good supportive evidence, but the parasite still needs to be definitively identified since antibodies to other nematodes can cross-react. Alas, PCR is probably the way of the future. The best treatment is ivermectin, which rose from an ignoble origin in a sample of soil from the Japanese golf course to become the treatment of choice for many helminth and arthropod infestations. By activating neuromuscular chloride channels, ivermectin hyperpolarizes and paralyzes the nematode's pharyngeal musculature, presumably causing it to undergo a slightly inhumane death by starvation. Luckily, these channels are sufficiently different from humans that we, too, are not paralyzed at the usual doses. 200 micrograms per kilogram once daily for 2 days should be enough, with follow-up stool and serological examinations for at least a year to ensure eradication. In her case, we would also hope that the eosinophilia resolves. Prevention of future infection is with shoes. Hope this is not way off the mark for my first try. I'm a regular listener, but have never before ventured out into the open. You guys have kindled in me a new interest in parasites and tropical medicine. Eternally grateful. Lachlan, medical student, Brisbane, Australia.
What a fantastic answer.
I
think,
think it's right, Dickson?
This is a medical writer of the future. I'm not going to say right or wrong because we don't have enough information to judge yet.
What about Mancinella?
Mancinella ozarti?
What do you think? Or Mancinella perstans?
Yeah, that's another one. Or Streptocerca.
Common in these countries.
It is. That's correct. Mm-hmm. And
eosinophilia is a consequence.
It is. So what would you do next then, Vincent?
I would do a PCR for Mancinella perstans.
If you had one, you would do one. That's true. But given a standard laboratory diagnostic procedure, what would be your guess on this?
What do you mean?
I'm trying to figure out what the next test is because we don't have a clue as to what this is until you do some more
We
testing.
don't have any labs. All we have is eosinophilia.
I know that, but Daniel does have a lab, and this woman has returned to a place where there are laboratories available. So I presume that we have a definitive answer. So we're going to have to actually probe.
But she is asymptomatic.
That doesn't matter.
Why did she seek medical attention? Was this an incidental finding?
Yeah, they do these post trip
I see
examinations before you muster out of the Peace Corps because very likely she did pick up something that could be treated at that moment. But we're going to have to now ask for the proper tests.
A shoeless peregrination.
Peregrination. I'm not sure you can do that in public and get away with it. I mean, peregrinating in public, I'm not sure.
A journey, especially a long or meandering one.
Exactly right. And it's like a walkabout. And they're from Australia, and walkabouts in Australia are very common, especially among the indigenous peoples. And in fact, I did several of those myself while I was there.
All right. Well,
you did those in public, right?
I did those in public, and I did them along rivers mostly, but
okay.
I peregrinated along the river.
Well, what shall I say? I mean, one of the things that I think is nice is when you get an individual with eosinophilia to go through the differential in your mind.
Sure. Absolutely.
But we sort of have a little bit of a cheat here, right? We are on this week in it's parasitism, right? Not parasitology, but parasitism.
Exactly.
So there's a bit of a hint. In the future, maybe I'll throw someone aboard with an autoimmune disease or someone who's taken a medicine that is causing the eosinophilia.
Or has a rash that unexplained.
Exactly.
Sure.
So in this case, we know what, I should say I have here, what was the definitive test, but I like the differential. I'm going to go down actually to Lauchlin's. He went through a differential here, right?
Yeah.
Where he starts to think about, okay, if we're going to think about it, helminth, it's going to be one, that would be invasive. He does also list that there are a few protozoans, and it's nice to have these memorized, I think, for test taking.
Can I just stop you 1 moment? Are you sure that Lachlan is a male?
Not actually. Do you have any idea?
No, we don't have an idea. So we actually got called on the carpet for this on TWiP. So I would hesitate to call he or she. This is the medical student Lachlan.
And these days gender is not binary anymore.
It isn't. No, it's
so. Why don't we just say Lachlan?
That's right.
Yeah. No, I guess we have to.
I'm sorry, but I've become sensitized.
No, I think that. No, I actually appreciate. I know a Lachlan, a Scottish guy, and it was a guy's name in Scotland. But maybe
you could be right. You could be right, but why take a chance?
Why do we have to?
According to Wikipedia, Lachlan is a masculine name of Scottish origin.
Okay,
but of course, my son's name is Devin, but many women are named Devin.
That's right,
and many others.
My older sister is named Dwayne. And
cool.
The Dwayne is a male name.
That's a cool name for a woman.
It is actually,
Yeah, no, and actually, I'll say this as a positive thing. He living in Port Washington, right east of New York, the people are, I will say, progressive, and they're even at when you go to a bathroom at the high school here, you have a choice. You don't even need to sort of make a gender identification. You could identify as a woman, and there's a room. You could identify as a man. There's a room, and then there's a sort of a binary option, and it's just a bathroom, like a gender neutral. So, I don't know, Lachlan, whether you're male, female, gender neutral. I still,
sorry I brought this up.
No, actually, interesting. When you go to do these trips where I leave the United States, it's very interesting when you start thinking about sort of the cultural context that we take for granted.
Right.
And gender identity is, I think, something that a lot of people don't even think about. Yeah. And then they get maybe, I think, sort of upset when the gender identity norms are sort of challenged and annoyed. And but, people on the other side who are not comfortable being forced into a certain, identity. It's nice, I think, that society is becoming more reasonable, flexible, gentler, kindler, etc. So, Lachlan. In the email from Lachlan, the different protozoan are brought up: Cytoisospora belli, Sarcocystis, Dientamoeba fragilis, and those would be the three that we would mainly think about. There's the ectoparasites, and actually some really severe ectoparasitic infections I saw this last week, to the point with scabies, for instance, where I would be surprised if they did not have eosinophilia, with just the severity of the infections.
Sure.
But in general, the rule is thinking of tissue invasive helminths as being the main cause of eosinophilia. I think we've talked to people about how in malaria we worry about eosinopenia, suggesting that they might be septic, a clearing of the eosinophils, and actually maybe a secondary bacterial infection. And then the next thing I like to do is, once you have a list in your head of what might potentially be there, is in a geographical location, what are the common pathogens, and then we could jump to Wink, who points out that Onchocercovolvulus, Loa are quite common there. He talks about how you could think of a Bancroftian filariasis, but not particularly common in this geography. So the geography is helping you. and to just nod our head at Joseph, is he quotes up to date where he says, 50% of the time you never figure this out. I think that's true maybe if you're looking at someone who never left an industrialized, developed country.
Right, exactly.
But if you actually say people who spent 2 years in West Africa working in the Peace Corps with eosinophilia, I think it changes the numbers significantly.
Yeah, they probably get more persistent in trying to find out what's causing this problem, even though it's not a real problem, but they know they have it, and therefore they perceive that it's a problem because they've been to an exotic place.
Yeah, well, once we actually sort of unveil what it is, maybe we could talk a little about like how important it is to know the diagnosis or not.
Absolutely, no question.
Let it go.
Yeah.
So, not knowing, I'm going to cover my answer for a second so I can, one of the first things I probably would do, as suggested by people, serologies, stool examination for over and parasites. In today's world, the nucleic amplification testing, which gives us a really broad catch. But in this case, a very interesting thing was done, which one of our writers suggested, which was blood was sent for an evaluation. And even without concentration, they saw something. They saw these really small filaria, microfilaria,
nice,
with a sheath and 3 terminal nuclei.
Oh my goodness!
And they were actually identified as Loa microfilaria.
Nice. Cool, very cool.
Yeah.
So that means that somewhere in her body, in her subcutaneous tissues, she's got an adult worm, several.
Yeah.
Which means that someday, and I know this is gender specific in this case, the only one that I know of that happened here at the medical center in New York City was I was sitting at my desk, of course, waiting for them to call me. I was doing something else, and I got a call from the ER, and one of the residents there said, Dr. Despommier, we were given your name by one of our fourth-year medical students who's now working with us, and he said, to call Dr. Despommier, he'll know what it is. And they said, there's a gentleman down here from Ghana, and he's just told us that there's a worm in his eye, and so I'm calling you to find out, A, is that possible? And B, then if it is, what should we do? Remember everybody out there, I don't have an MD degree, but I know what to do. Okay. So I said, A, it's very feasible that someone from Ghana would have a worm in their eye, especially when they were looking in the mirror shaving that morning, which is why I use the gender specific in this case, because this woman obviously wouldn't catch her worm that way. But certainly he was able to see that this worm has migrated from the subcutaneous tissues, let's say in his arm or his shoulder, up through the neck region into his cheek and then across the sclera of his eye. And then he looks in the mirror and there it is. He knows the history of this. It's painless. He goes to the ER and says, Doc, I've got a worm in my eye. Would you please take it out? And it sounds like the start of a joke, But obviously I said, you can look up the procedure here, but you can anesthetize the area of the eye on both ends of the worm and then make a small slit with a fine scalpel. And the worm will probably end up crawling out of that slit, and you just grab it and pull very slowly, and out comes the worm. And that's how they take these worms out. And they're quite big. I mean, they're maybe 3 or 4 inches long. They're not very thick, thank goodness. But that's the procedure. And the doctor was absolutely stunned that it's so easy and so well known in some areas of the world. So maybe our former Peace Corps worker has something like that wandering her body, and maybe she's got calabar swellings, and you could ask about different things. And I hope I'm not preempting anything you're going to say here, Daniel, because that would be my next move once you've made the diagnosis, is to...
Yeah, no, I actually think this is, we're starting to, and maybe we should just really, as we've suggested before, talk about the life cycle and how did this happen? So,
did they find any other microfilaria that didn't have 3 nuclei that went out to the tail with a sheath? Because it's possible that you could have 2 things. Right?
It definitely is.
And then remember what the contraindications are. If you found, let's say, Wucheria bancrofti and Loa together, then you've got a problem.
Yeah, or Onco-Circa, Loa, and I think we had a paper where we talked about
exactly
so treatment issues are related to, whether it's mono-infection, levels,
right.
etc.
And did they look for nodules under her skin by doing a palpation?
So she did not have any other nodules, or she did not have any noted nodules.
Okay.
But maybe should we talk about the life cycle? Because I think that's really interesting with the time course here. And one of the things, this is one of those parasitic infections which a person can acquire from a vector.
Yep.
And how do we remember lower? Like what is like what gives it to us? And the way my little mnemonic is, it's almost got a pet name, Loa.
Right.
So I'm not sure what Dickson's doing there with his hands, but it's very
making we'll get to that. I'm imitating the vector's mechanism for getting blood out of a victim.
Actually, no, that is good because we'll get to. So it actually comes from the deer fly.
Right.
There's several other names, but it allows me to think Loa is one of these affectionate names that maybe to intimate people might, sweetie or something. And, because I'm always trying to memorize the vectors. You think I would just know them after a while, but I always like to think of memory tricks for people just sort of coming into the trade. The idea with, river blindness being caused by the blackness fly or the black fly. Yeah. But Dickson was actually making a good point is with his hands, which is that it's not really like a mosquito, which is going to really have almost a hypodermic. There's going to be a cutting. They're actually going to make a wound, and it's through the open wound that the larvae crawl into the host.
That's right.
So it's starting there, and then there's actually quite a gap. There's really a period of time from the time that the larvae invade the subcutaneous tissues to the time they develop into adults. And it's years. It could be 1 to 4 years before you can potentially have the production of the microfilaria. So right out of Peace Corp, you could have you could have a window where you're not able to make this diagnosis in the blood.
Exactly.
And then and then once you get to that point, and then you actually, can potentially have the worms mate, the females begin depositing the microfilaria, and then you're going to see the microfilaria in the bloodstream.
Exactly.
But what if we just, and then once through the bloodstream, you can repeat the cycle, right? The fly can take its blood meal, it can ingest the microfilaria. They can develop to infective stages in the fly, and then someone else gets sliced. But what if we say, you know what, you got these, it's fine, just give it a few years until those adult dies, because the adults, they only live for up to 17 years.
Wonderful.
A mere 17 years.
Now, the young generation might find that to be a long time.
That's right. That is exactly right. It's remarkable how long they do live, because obviously they are an invader, but they have their own little mechanisms for inhibiting your immune system so that they're not attacked, and yet they still leave their antigenic signature in your subcutaneous tissue, which induces this swelling response behind where they are, and that swelling follows the track of this worm as it migrates through your tissues, and so there are immune responses, but it doesn't end up in protecting, nor does it end up killing the microfilaria. Now, I'm blocking on this one because I don't have our textbook sitting in front of me, but I bet you Daniel does.
Well, the only time I separate myself was, for instance, when I was in Panama, because it is heavy.
It is heavy. We get the PDF version. So does it say whether or not the microfilaria of Loa have Wolbachia?
Oh, so that is and I actually made this comment to you early on. Loa is unique in never having Wolbachia identified as endosymbionts.
Okay, good. Well, we just happen to have another hard copy of our book right here.
Okay.
And we can definitively say this or not, and I'm sure that we are correct.
Do you want to hear the sentence?
I want to hear the sentence.
Unlike other filarial nematodes, there's currently no evidence that Loa contains Wolbachia symbionts, based on extensive investigations employing light microscopy, electron microscopy, and PCR.
Bingo.
Okay.
That says it all.
Yeah.
That's wonderful.
But how did you treat the young lady, Daniel?
So I actually like I quoted this. She was treated with DEC, and she became hypotensive for about an hour. But eventually, things turned out well, and she was parasite free.
So what is DEC?
So are you're going to make me pronounce that? Isn't that your thing?
It is, actually. It's diethylcarbamazine.
Exactly.
Diethylcarbamazine. That's right. Which has some risks in taking the drug, by the way. I mean, it's not symptom free. There are side effects from this drug, unfortunately. And for many years, it was the only chemotherapeutic agent that they were using to treat filarial infections with.
And this is because it's readily available, not ivermectin?
It's cheap, it's readily available, and it was effective, but it also had some side effects.
But ivermectin has no side effects, right?
That's correct.
It's amazing.
It is an amazing drug.
It's amazing how well tolerated ivermectin is. But I would say well, two things I'll say. So as we say in our textbook, ivermectin is not a preferred agent. In saying ivermectin has no side effects, Loa might be the exception. Is there may be an issue with the destruction of the incredible high levels that sometimes you can see in these people? Yeah. So what I would recommend, I'm not sure that in all honesty, if you see blood without concentration that you want to just hit somebody with DIC, what we usually recommend is you try to lower those levels below a certain threshold. It's about 2,500 microfilaria per milliliter. And you could do that either through apophoresis or, in all honesty, daily albendazole for a while. And then you wait till you're down, and then you do the DEC. And at that point, it is well tolerated. It's pretty safe. And
so remember that the ivermectin only affects the production of microfilaria. It doesn't actually kill the adult worms. And that's why they had to give it for so long for Onchocerca, because the worm can last for 20 years under your skin, just like Loa Loa. Although I don't think Loa Loa lives for that long, but 17, but who's counting at that point, right?
Yeah, but I would say this will be one of those, maybe we throw pearls every so often, but there have been cases where the microfilaria is above 30,000. In that case, there's been an ivermectin-induced encephalopathy.
Oh my goodness!
Right. So we normally think, just throw ivermectin. It's all good. Don't worry about it. And this, I think we discussed in one of our papers. It was the Gleaback paper, right? Oh, right? Is that we're looking for other options, because if you're throwing ivermectin at people with onchocerciasis and unbeknownst to you, in your group you have someone with high levels of the Loa, you can actually end up, causing some pretty significant morbidity.
Wow. Meanwhile, she's symptom-free. I mean, this poor woman, all I have is my raised eosinophils. What's wrong with that? Why are you giving me this drug to begin with, right?
Did you see the eosinophils go back down after treatment?
Yeah, good question.
And that's actually typical, is that once you treat them and they clear, then the eosinophils would go away.
Yeah. Yeah.
And this is, as one of our emailers pointed out, this is not something where the adult worms are going to be able to then have the microfilaria and complete the life cycle. This is just going to happen, and then the only option for the parasite is to be taken up by a fly, where it can then transform back into an infective state. So this is time limited. It's only going to last a certain period of time. The worry might be, as Dickson describes, is when someone starts getting swellings because you're getting migration through tissues, or the upsetting, I should say, aesthetically displeasing event where a worm crawls across the eyeball.
Yeah. Yeah, there's a ton of pictures out there on that particular event, and showing the worm, and it's...
Oh, yeah, many. And we have just seen them in your new video.
It's true.
And we did a TWiP on it many years ago, and I. That's right. Doctor, there's a worm in my eye. That was the name of that episode.
That's right.
So yes, we're very familiar with this.
Yeah.
So in the future, we'll be able to refer people to the videos, won't we?
We will. We will. So we're developing an online course, is what Vincent means. And indeed, we will have video portions of that as well to show some of these things live and up close.
45 videos.
Yeah, with all three of us. You're the producer,
well, not
obviously.
me, but you and
Daniel and I are sharing the responsibilities here, and it's been a lot of fun, actually.
Behind the scenes.
Well, I'm going to throw one more thing in, and it was someone made the mention here of Eck Bombs Syndrome, and there was actually a colleague of mine here in the New York area, And she identifies as a female. She was describing a case where a poor woman had been sent to multiple doctors, and they had all just said, "You've got delusional parasitosis." And what she actually did is she had these migratory swellings from loa.
You're kidding.
And she was finally diagnosed. But unfortunately, and this one was from Nigeria, and she'd been, in sort of, I guess, several neighboring countries as well. But when she went to different doctors and described, I have these swellings and they seem to move, and they just said, "You're crazy." And unfortunately,
it's too bad.
It's I think embarrassing for the profession when we label someone as crazy when really we're the ones who have the sort of educational deficit.
If you don't do all the tests, then how can you say that she's not infected with something? Then you have to do the tests first, and you have to persevere.
Yeah. All right.
Very good. Thank you very much. This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone, including you, Dickson. I know you're a good chef.
Okay.
So I was just kidding, because I know you don't need to learn from Blue Apron, but you could benefit by tasting it.
I could.
Accessible to everyone while supporting a more sustainable food system, setting the highest standards for ingredients, and building a community of home chefs. For less than $10 per person per meal, Blue Apron delivers seasonal recipes along with pre-portioned ingredients so you can make delicious home-cooked meals in 40 minutes or less. You can customize your recipes every week, so you can customize them because you would want, some people don't want certain things, right?
That's true. Or they want extras that are not
That's called dietary preferences.
Yeah.
And there's no weekly commitments. You get deliveries when you want them.
Mm-hmm.
They have a freshness guarantee that says everything you get is ready to cook. And if not, they will make it right. And they ship exactly what you need, so there's no food waste.
Sounds great.
And that's a big deal, right, Dickson?
It's a very big deal.
Here's an interesting new twist. This October, which is the month we're in right now,
yes,
Blue Apron is celebrating its 5th anniversary. They are bringing back its top 20 recipes.
Oh, wow.
And these have been picked by the Blue Apron community. This is for a limited time only. So if you harken back, is that the word? If you harken back on a recipe you had a while ago, this is the time to order. Blue Apron is all about giving people fresh recipes to explore as they learn to cook new dish after new dish, which is why they don't normally repeat cycles within a calendar year. That's why this limited time offer is so exciting, because you can go back to the ones that you really liked.
Amazing.
Now, have you ever tried Blue Apron, Dickson?
I have to be honest with you.
You didn't.
I could say no.
You didn't. Not. I haven't. Daniel has, of course, many other podcasts.
I like shopping for my food. I'd like to do that. That's part of the adventure of cooking.
You could not shop one night, and you could try.
I could not shop one night and have a Blue Apron.
But I like Blue Apron because I hate shopping.
I think you're right. You do.
Dislike shopping. I think supermarkets are.
There's a lot of people that agree with you. But,
weird places. Lighting is weird. I don't know. Anyway, so that you get delivered to the door, and they're really good. I'm telling you, I'm not a great cook because. Why? Because, I don't know, I just am impatient, I guess. But,
good in the lab.
30, 40 minutes. Yeah, isn't that interesting? But I don't have to eat what I make in the lab, I guess.
This is true. This is true.
Anyway, you can try Blue Apron's all-time customer favorites. Go to blueapron.com/TWiP to get $30 off your first delivery and free shipping. blueapron.com/TWiP. If you go there, it will say, listeners of This Week in Parasitism. How about that?
How about that is right.
You'll love how good it feels.
That's right.
And tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron is a better way to cook.
Nice.
It's really amazing. And that you could even imagine doing this, right?
It's true. Especially if they can guarantee it's parasite-free. The whole world would flock to them then.
Well, I don't know how you could do that, Dickson.
Well, I think you do this by carefully selecting your purveyors so that everything is fresh.
Purveyors?
Oh, yeah. That's a very tricky subject.
Tricky, very tricky.
It's tough to pick out. And sustainable, that's the other thing that's very tough to get is a consistency of quality.
These are the buzzwords: sustainability, responsible
that's right.
farming, right?
Yep. absolutely. And they're very conscious of that. So I have deep respect for them, even though I haven't tried them.
We have a paper now.
We do.
It's from PLOS Biology.
Indeed.
And it's called Extreme Genome Diversity in the Hyper-Prevalent Parasitic Eukaryote Blastocystis.
Right.
And there are a lot of authors on this paper. We have, let's see, do we have co-first authors? Doesn't look like it. We have, first author is Eleni Gentikaki, and the last author is Andrew Roger. These are from Dalhousie University, the University of Ostrava, which is in the Czech Republic, the University of Alberta, CNRS in France, King Abdulaziz University.
All over.
Université Côte d'Azur, or Côte d'Azur, not Côte, Côte d'Azur in France. We have more, University of Exeter in the United Kingdom.
Right.
The Canadian Institute for Advanced Research, the London School of Hygiene and Tropical Medicine.
Listen to this.
And the University of Exeter again. Wow.
Yes.
This is a big thing. Now, Dickson.
Yes.
The first sentence in this paper, Blastocystis is the most prevalent eukaryotic microbe colonizing the human gut.
I think they're right on that.
1 billion people.
1 billion people.
Where have we heard before of Blastocystis?
We've mentioned it. We've had it in parasites of minor medical importance.
Have we done a TWiP on it?
We must have.
Is this one where someone was sick and they had this, but they could never prove that this was causing it?
It was. This is still seeking verification as a genuine pathogen.
Desperately seeking verification.
And maybe it's because of this.
Daniel, have you ever encountered Blastocystis?
I have actually, and I was gonna read a sentence from this really nice red textbook that I have a copy of.
I have a similar one.
This actually came up when I was doing the Peace Corps talk. It was where they had all the medical officers from different Peace Corps stations, and they had a bunch of cases that I was going through. And one of the questions came up. It was really the question is this a pathogen? And so our sentence, our the way we address it, we say, several cases have been described that defy any interpretation other than illness caused by B. hominis, based upon an extensive negative laboratory finding regarding all other known pathogens of the GI tract.
Right.
But yet, its pathogenicity remains controversial.
It does.
I say it's really tough when something is so prevalent that a billion people, you can identify this in their GI tract. And then most people, and I do mean most, out to several decimal places, are doing absolutely fine.
Correct. So that's why I picked this paper. I actually discovered this paper by accident, but the moment I saw it, I knew that we had to do it because, as we will see, and I'm sure it will reveal the breadth and depth of this infection. It ain't the same thing no matter where you look. This is the most diverse group of the same genus organism that infects humans of anything we have knowledge of.
TWiP 54, Unde Venus.
Ha-ha.
Vincent and Dickson consider a case history of a young man with Blastocystis hominis. Is it causing his disease? That was May 23rd, 2013.
Goodness gracious.
That was in the old days, Dickson.
That was way back.
Well, that was before me. That was the pre-Griffin period.
Pre-Griffin.
That's right.
Pre-Griffin.
Pre-MD verification. I think that this article, because it has so many authors, I think there are so many authors because they were clever enough to realize that since they found so much diversity, even in the small sample that they had access to, even one author, was they started to ask around, they got samples from all over the world, and then they really began to make sense of all this story. And this is a very interesting story, by the way. Fabulous.
There are at least 17 subtypes.
17. Count them. 17.
And they've been isolated from various mammalian hosts.
Right.
And 9 of those are from humans.
Yes.
And so this is one of the subtypes that they've obtained here. This was from a symptomatic male.
That's right.
Of course, we don't know if the symptoms were due to blastocystis or not.
We don't.
But this has been sequenced, and they have a few other subtypes that have been sequenced in previous years, and so now they can compare them and start to say what's going on here.
Exactly.
To me, what's amazing, first of all, the genome is 16 megabases, 16 million bases.
Right.
And the others are different. They're slightly different, but not much. But the number of proteins or protein-coding genes, 6,500. Now, the human genome contains maybe 4 or 5 times as many, but our genome is 3 billion base pairs.
Correct.
And of course, I guess that's what you need to make 2 arms, 2 legs, 2 eyes. I don't know.
We have a lot of endogenous virus sequences.
We have a lot of
to put
stuff
up with, right?
at Blastocystis. They call it several times here a very compact genome.
Compact. Exactly.
Compact genome.
Exactly.
So do you think we should call this episode Blasting Blastocystis?
Well,
because BLAST, of course, is one of the computer programs you use when you identify.
Perfect.
I
That's
like
perfect.
that.
I do
actually.
too. I do too.
You know, I will say when I was with Floating Doctors, one of the volunteers there is a fan of TWiP, was very excited.
Nice.
Cool.
And one of the things that she commented about was our clever titles.
Okay, well, Blasting Blastocystis.
You should try TWiP, man. Talk about
TWiP is off the wall, off the
Daniel,
charts.
one of the recent ones was called Pork and Jeans.
Okay,
Isn't that great?
I like
Jeez, yeah.
that's
Alan. Alan is a genius at those titles. Absolute genius.
So let's see. What do we have here? When you compare, GC content is quite variable.
Quite
Among the 3 different subtypes, ST1, which they did in this paper, and then they have ST4 and ST7 previously. The GC content ranges from 55% to 40% to 45%. That's really remarkable.
I would say.
In fact, they say at some point that these could be different species, really.
Exactly.
And maybe they are.
They don't even commit to a species. In fact, at the end of this paper, they say there is no such thing as Blastocystis hominis.
Yeah, that's right.
You just got to call it something else.
So the GC content varies by 15%. The gene numbers varies hugely.
Right.
Hugely. I mean, it can't be the same species.
What's going on?
It's ST7. ST1 is more compact. Very, very much fewer distance between genes.
All right.
So lots of little things like that. Now, the one thing that's very cool, and I had not heard of before, is polyadenylation-dependent generation of termination codons. So, gentlemen and listeners, what are the 3 termination codons in messenger RNAs? Does anyone? I know I knew this, but
AAT?
no. UGA, UAA, UAG.
Oh, okay.
Okay. And you notice that you could, let's take UAA. So let's say you have you're making an mRNA and you get to this U, and then the polyadenylation, if added there, would generate a UAA, would make a stop codon, right? So that's what they mean by polyadenylation-mediated stop codon generation. It's not in the DNA. The stop coding is not in the DNA. It's made by polyadenylation.
How interesting.
Isn't that cool?
Yeah, it's very cool.
They say this is unique amongst eukaryotic nuclear genomes.
I was going to say it's not neat. I found it slightly creates a challenge for us because it's very hard for us to just read the sequence and know where the stop codons are because now you've got to add this other layer of
And in fact, they say this makes it difficult to know how many protein coding regions there are in blastocystis.
So they need to do a proteasome next, and they need to see how many proteins there actually are.
Proteome.
Yeah. They can culture these, by the way, so they can get clonal varieties of them.
As
so
they
that's
say,
good.
most gene-finding algorithms, and these are computationally based, right?
Yeah.
Are predicated on the presence, as Daniel said, of a stop codon. So you can get 15% of ST7 genes have stop codons generated by polyadenylation.
Yeah. A big trouble with this organism, however, is that it's an obligate anaerobe.
Obligate anaerobe.
So you have to be careful with this one in the way you grow it.
Yeah, so air will poison it, right?
It requires special equipment. It's
not even micro. Aerophilic.
Nope. And it's related to algae and fungi.
Is malaria an anaerobe?
No. No, it's not. It's got a modified mitochondrion. This one does too, by the way.
It does have an interesting mitochondria.
Very interesting mitochondria, but it doesn't derive its energy the way we derive energy from air and use that as an oxidizing agent. This organism doesn't care for that at all.
So one of the other differences is that this genome is more intron-rich than the other two, but they say it could be just methodology because, you use a computer program to do it, so they say we don't know if that's real.
It has no heme proteins.
Has no heme proteins. Oh, before we get to that here,
Sorry.
The extent of dissimilarity among the subtypes supports the contention that they should be considered at least equivalent to separate species. Blastocystis Dickson, Blastocystis Griffin, you could all do that.
That's right. Vincenzo. We'll have Blastocystis Vincenzo.
There are 221 kinases encoded in the genome.
Incredible.
Did you know there's a website called kinase.com?
Did not know that. The kinome.
All the kinases.
There's a kinome.
There's a kinome. All the kinases are part of the kinome. Isn't that great?
Ohms for everything now.
We have a Dicksonome.
No, we don't.
All the Dicksons in the world.
No, they're sitting right here. Thanks.
And they don't know what these kinases do, of course, but now we know they're there. There are 89 secreted proteins.
I was going to come to that.
Do you like that?
I love that. Because
they're all predicted, of course, remember.
Of course, but this is where the pathogenicity comes in, right?
Yeah, they talk about other parasites that have.
Some do,
for example,
some don't.
There's some secreted collagen-like proteins, and they
That's
say
weird.
In other pathogens, these have been implicated in pathogenicity. So if you really wanted to work on this blastocystis, you could start looking at these genes. Is there an animal model?
It has no salvage pathways. It can make everything de novo.
What does that mean?
Nucleic acids.
Oh, yes. Yeah, it makes everything de novo. That's unusual, isn't it?
Very unusual.
And also,
there's a lot of drugs interrupt the salvage pathways.
Right. So you could target it for this thing that's not been associated with any disease. You could make drugs against it, right?
Exactly right. But in this case, if you make a drug against the de novo synthesis of these, you're going to kill yourself at the same time.
Then here's one that you would like, Dickson. The ATP generation is coupled. These guys typically couple ATP generation to the production of hydrogen and not oxidative phosphorylation. Hydrogenosinase.
Hydrogenosomes.
Hydrogenosomes. That's what I meant.
So name the other organism that has hydrogenosomes.
Yeah, Christian de Duve.
No, it's Trichomonas vaginalis.
Trichomonas?
Yeah.
They make hydrogen.
They do. Miklos Mueller actually worked with Christian de Duve on that one.
And so if you light a match, it will explode over.
Or if you want a source of hydrogen, molecular hydrogen, which is...
Do we want a source of hydrogen?
Absolutely.
For what?
Are you kidding? We can drive all of our powered supplies by hydrogen-powered
So
engines.
we just culture some blastocystis in the tank, right?
Or clone the genes for just the
better.
hydrogenosome, and then you can just do that.
Huh.
Sure, absolutely. Remember, Craig Ventnor went around the world looking for organisms that secrete hydrogen.
Hydrogenosomes.
And here are two of them right here.
So they look at proteins that are putative members of these hydrogenosomes, right?
Right.
Which they put a lot of time into this. They have a nice picture showing
a lot of time
these components.
I ran across a word. I'm not sure whether either one of you knew doing this before, but stremenophiles.
I don't know. I thought you would know that.
Yeah, well, I had to look it up. Actually, it's a category of fungi.
So you know,
it's an unequal length.
Stremenophiles.
Yeah, they have two flagellae, but one is longer than the other. And it's quite a different group of organisms I'm totally unfamiliar with.
Yeah, but not all have flagellum.
No,
for instance,
no,
our
that's
guy
right.
Here doesn't. There's two
Exactly.
lines. And why would an Irishman be aware of stremenophiles? Because it's actually in this group that the cause of the Great Hunger, the Great Irish Famine.
Yes,
right. So that's
right.
Maybe that's going to bring back a memory there.
A fungus that killed off the potatoes.
Now these guys don't have any peroxisomes.
None.
Dixon, what is a peroxisome for?
Well, I think it's to counteract superoxide dismutase buildup products for oxidative damage to tissues.
Are you guessing, or you know?
No, I think that's what I remember from the time that I was working over.
Yeah, but it's a long time ago. It could be changed now, right?
Could be changed. And in fact, you're probably changing it right now as we speak.
No, I'm not. I don't know.
No, I think peroxisomes contain all the enzymes which counteract the buildup of oxidized material in your tissues.
So,
Maybe I could say they're for breaking down long-chain fatty acids.
Got
Would
it.
that work?
That's
Can I throw that in?
All right.
That's one thing.
That's fine.
Yeah, fatty acids. Two functions: break down fatty acids and transfer hydrogen from compounds to oxygen to create hydrogen peroxide, and then convert hydrogen peroxide into water.
So
water.
That's
New Jersey
disarming oxygen.
So, which is why in humans there's a cell that has a lot of this, and it's that's why the cells are white.
Got it.
That's a joke. But anyway, it's common in a lot of our immune cells, right? Because it's basically used in defense. But maybe that helps people remember that, because the peroxisome is often used as a way of combating microbes.
Got it.
And they go through a lot of other gene groups that are present that they see.
Some weird stuff, though, right?
Not a lot of cytochrome P450 genes.
It says none. It lacks all genes encoding heme-containing cytochrome P450 protein. So how does it detoxify?
I don't know. That's a good question.
Probably uses hydrogen as a reducing agent.
So T. gondii has a single cytochrome P450 gene.
Yep. And they compare it to Giardia, which is the other anaerobe that's culturable that lives in your gut that's a protozoan. And its metabolism is totally different than this one.
Yeah, they say that since these genes are involved in sterol metabolism, and since they are essential for eukaryotic membranes, the lack of cytochrome P450 in Blastocystis suggests that it obtains sterols from its host, as does Giardia.
There you go.
So anything else that struck you, gentlemen, is remarkable in this?
Well, I mean, outside of the number of variants of this organism, that's enough to get you started on anything.
It does not answer the question, though. Does Blastocystis?
Is it a pathogen?
Is it a pathogen?
Or if it is, which one of these multiple varieties is the actual guilty party? If there's just a couple, maybe that explains why we are so unsuccessful at pinning down what's wrong, because maybe just a very small subset of this causes disease, and all the rest of them, although they're present, don't do anything.
Oh, here's something we forgot.
They
found it has a near-complete meiotic toolkit.
Yes.
Possibly indicating a sexual cycle.
That's correct, although none has been described yet.
None has been described.
But this also is capable of encysting. So it has some unusual...
No, I insist.
Yes, you do. I insist, said the amoeba.
I insist. Give me the check.
The flagellate says, of course you do. Yeah, that's weird for an algae or a fungus to insist. So it's got some protozoan-like characteristics, and it's got some plant-like characteristics. It's a sort of a hybrid. And it's causing people to, really second-guess what they know and what they don't know about the microbiome of the gut tract, for instance.
In conclusion, this study provides an important window into the biology of blastocystis, and that can guide future experimental investigations. I can say one thing with certainty. The future is hard to predict, Dickson.
Yes,
but I can say one thing.
Please.
Dickson will not work on blastocystis.
You can say that for sure. That is exactly
And I'll bet Daniel will not either.
Probably not. No. But I did think what they did was really, well, again, like to talk about things like Ekbomb Syndrome, where you have people coming in and maybe they've got diarrhea and symptoms. You say, you know what? All I'm seeing is this blastocystis, and it says here in my book that's not a pathogen. Go on your way. And I think what they're saying is, we don't know everything. And wow, there's a lot we don't know.
Sure.
And this blastocystis hominis may be such a huge bucket that a small, maybe subtype, maybe this ST1, and then maybe only certain types of ST1 in certain individuals are pathogenic. So it really opens,
that could be looked at, right?
It could be. And then you've got the other problem of, okay, it's a pathogen and we know it's causing the problem. Now what do we do?
Are there any drugs that will inhibit this organism?
No.
Maybe we should get some, but then there's so many different types. Maybe you need many different ones.
Which metabolic pathway would you attack?
Maybe it's normal flora. Maybe it's a symbiont of our gut
that's
tracts.
right. Exactly. If you don't have it, maybe something goes haywire. Who knows?
When you treat people with antibiotics, let's say, before gut surgery, give people,
Yep.
and you wipe out the microbiome, do you wipe out Blastocystis? Probably
No,
not.
you don't. No, you can't.
It's still there.
It is still there. That's correct. Maybe amphotericin B would be the way to go, or something else that attacks fungi, but it's certainly, assuming it's a protozoan, doesn't help you with the chemotherapy.
Yeah. All right. Daniel, did you have anything else you wanted to mention about the paper?
No, actually, I think it's a great paper. I think it really is humbling, and it raises this whole issue. I guess I'll just throw in the last thing. There are some potential treatments, and go too far down this road. But when someone has symptoms, they have a heavy load. Let's say they have just many of these, and you finally say, well, let's treat and see if there's a therapeutic impact. You could use some of the flagellate targeted medicines, things like metronidazole, tinidazole.
That goes after the anaerobes.
Yeah, some of the anti-folate metabolic things like Septra, Bactrim, the sulfur drugs. And then nitazoxamide, which we're starting to use more for resistant Giardia, gets thrown in there. Paromomycin. So there are some options out there that people can try should they get to a point where this is all that's identified, and as we sort of say, the clinical context sort of defies another explanation.
Right.
All right. Now we do have a case report, and this is something different. And I think Daniel should present it because historically he has done this.
I totally agree.
So Daniel, tell us about this.
Well, this was sent in by one of our listeners from Kenya, and I made a few slight little modifications so that people wouldn't get it immediately.
Yeah, yes,
right.
Yes,
because I want people to say like, oh, you told us the case and what it was. So
that's very low hanging fruit.
Yeah, I think people will get this, but what they might not get are some of the subtleties that we'll end up discussing next time. They may say it's such and such, so we're going to raise the bar on this one. And this comes from a longtime listener, and he is describing a case. Not only is he describing it, he sends us videos, which maybe we should save till next time.
Yeah.
And the case is a 1-year-old girl, and she's brought in by her mother with skin lesions that developed a week prior. There are 5 lesions in total. They're on the forehead, the neck, the back, the chest, and the right arm. We actually have a video of the right arm that will later be shared. The lesions look like boils or furuncles. So what do we mean by that? That goes back to I think the history of how we used to describe staph skin lesions. And it's sort of almost, a fluctuant feels like there's not just an indurated or hardened area, but it almost feels like there's a sac or abscess type feeling. But the key thing is these keep changing in size and appearance, and there's a sensation of movement under the skin. Not sure how you get that history in a 1-year-old child, right? But that's what's described. They're painful, they're itchy. The child is breastfeeding well. There's no other complaints. Little background on this child. They were born at a county referral hospital. They got their vaccinations at a health center, which were all up to date in a rural health center area. The context, this child is living in a wooden house with a dirt floor. The roof is corrugated iron sheets. There are 2 rooms in the house, and the description is that they're actually sleeping not on the dirt floor, but on raised beds. There's a big community dam in the neighborhood, reported to have stagnant water throughout the year, a large mosquito breeding ground. They don't use mosquito nets. They do have a source of reliable, clean drinking water provided by the government. They have 1 dog, but they also have a neighbor, and they've got a dog, and it's coming over all the time. They hang their clothes on the clothesline after washing. They never dry their clothes on the grass, and the clothes are not hot ironed. The child is breastfeeding well, I think, as we described. No fever, doesn't look pale, no jaundice, not in any distress. There's a little bit of swelling of the lymph nodes in the back of the neck. These are tendile, a little bit mobile. And then, as we were told initially, there are these, call them pustular, furuncular type lesions on the forehead, chest, neck, back, and the right arm. And I'm only going to give you a little more description here. They're about 1 to 3 centimeters in diameter. They actually rise up a little. They are tender, and they have a central punctate area. And there's a little bit of drainage coming out. We have limited labs, but there is increased eosinophils, increased neutrophils, and there is a negative HIV test. And so, using the word "so" again, the couple things I want people to do is tell us what they think it is, but then I'm going to ask for a level of specificity. Don't just say, "Oh, I think it's this." Tell me, "I think it's this," and then maybe a little more detail on sort of what specific this it might be. Okay, and you can maybe use geography and tell us a little bit about why it might be this in Kenya versus something similar in other parts of the world.
And of course, since our emailer is not here with us, we can't ask him any questions as we normally would of Daniel.
Exactly right.
Yeah, well, I think I put everything right that I think we need to know from. This is from Dr. Arthur Mumelo, who sent us this case, and thank you. And we encourage other people to send in interesting cases. And the nice one, I like the videos because we really have a solid diagnosis here, which is going to be great for people listening and then writing in.
Arthur works in northern Kenya as a medical officer.
Exactly.
All right, we have a few emails to wrap up this TWiP. How do you like that? Wrap up this TWiP.
I like it.
Steve writes, and he sends a letter. He sends a link to an article in the ASTMH,
right?
Human Intestinal Parasite Burden and Poor Sanitation in Rural Alabama. And he writes, I expect that the TWiP team are already aware of this sad report on the return of once eradicated parasitic diseases with increasing inequality and poverty, but I thought I should pass it on just in case. Yep. Steve is from Luton, England. And this is, of course, about Alabama, which, as we know, has a lot of problems with hookworm, strongyloides,
that's right,
Nicator americanus.
Look at that.
What else, Dickson?
Just rolls off your tongue. Ascaris.
Cryptosporidium.
Giardia.
And these are not elsewhere as extensively in the U.S., are they?
Well, wherever you find poor sanitation, you'd find Giardia, that's for sure. And some of the other, like Cryptosporidium, you'd find that there too. But hookworm, no. That requires a longer time in the soil and the right soils. So he's right. I mean, that's a Back to the Future. This is unfortunate.
This study was done in Lowndes County, Alabama, because it has had previously high hookworm burdens, degree of poverty, and the use of open sewage systems. These are things that should not be carried on here,
totally
right?
Agree. Totally.
Because very sad.
It's tough. I was in Panama, as I mentioned last week, and on the way back, there was that whole big flight delay issue. Yeah. And I was taking a taxi from the domestic airport to the international airport, and I started talking to a couple that I was sharing the taxi with. And the wife, they were newlyweds, and the wife was saying that she works in an area of Tennessee trying to recruit individuals to that area, and was actually talking about the extreme poverty that is in the surrounding area, and saying that some of the areas of poverty in the United States rivals that really anywhere in the world. And it is amazing that goes on, and that from the economic side to even the sort of knowledge, you would think, my gosh, if you want to be an expert in parasitology, you probably just need to live in parts of Alabama or Tennessee, or it's troubling that here we are in 2017 and still in our own backyard we have incredible health inequity and economic inequity.
Correct.
The next
Peter.
One is about, I think, a previous case. Yes, please,
Okay.
Dickson.
Dear TWiP team, having looked at possible causes of ocular parasitosis in a teenage girl from New York, I feel that the parasite that fits this case is nematode Balis ascaris procyon. Balis ascaris infection is caused by a roundworm found in raccoons, larval migraines, results from accidental consumption of embryonated B. procyon eggs. After an animal or person swallows Balis ascaris eggs, microscopic larvae hatch in the intestines and then move into the bloodstream and tissues, causing damage as they grow. There is a significant risk that the worm seen in her eye is not the only one in her body. I found this recent article on Balis ascaris infection, and she gives a reference. And, yep.
I'm pretty sure Peter is a guy.
Yeah, I would say so.
But never know.
It's a wild guess, but we'll take it.
But we'll just say Peter.
That's right.
I think that was the right answer, Daniel, right, for that case?
It was, yes.
Yep.
It was. That's
Yep.
great.
All right,
Okay.
Daniel, can you take John's?
I can, actually. Hi, TWiP casters. I very much doubt this is the answer to the case study, but given the timing of the TWiP episode and the BBC News article, I thought you might find it interesting. And here we have a link to Thalassia calipadia. Actually, John writes, but then it says, thanks, Gareth. So maybe it's Gareth John. Maybe
Interesting.
his first name is Gareth, last name is John.
Or I might have just messed it up, you know.
Well,
Who knows?
Do you know anything about Thalassia, Daniel?
Yeah, it is another one of those eye worms. It is usually not in humans, but can be reported in humans.
Mm-hmm.
And I don't know if you linked on that.
Yeah.
But so it is actually another cause of eye worm, thalassioiasis, so another cause of eye worm.
Okay. The next one is from Dave, who writes, Good doctors. I was listening to Quirks and Quarks, our Canadian science show. Bob McDonald was interviewing a journalist who was with an expedition to eastern Honduras. The show aired August 26. Douglas Preston, the journalist, described going to Honduras to check out a lost city with a curse on it. It's a great story, but what interested me was the curse. It turned out to be mucocutaneous leishmaniasis. He, the journalist, went to tell how most of the people on the expedition caught the disease and that the treatment was almost as bad as the disease. Douglas Preston wrote a book about the adventure, Lost City of the Monkey God. It would be interesting to hear your take on the story and the accuracy of the effects of the disease. Mm-hmm. Thank you for the great podcast and all your efforts to educate the world. Shearer Dave from southern Alberta, where it's finally raining, woke up this morning to 1 degree Celsius, high today 8 degrees C. Does anyone know about this story?
I do not.
The Lost City of the Monkey God.
I have not heard of it.
I actually have that book.
Really? Who would have guessed?
It was a present from my mother, which I have not read.
You haven't read it yet?
No, but it is sitting on the shelf.
Let us read it together.
We should read it, and then we can answer Dave.
Let's have a book club.
Is the treatment for leishmaniasis as bad as the disease?
The interesting thing, when you read this initially, I was doing a lecture when I was down there in Panama on the different types of leishmaniasis. And we talk about there being three main, we try to keep it. So there's cutaneous, mucocutaneous, visceral. There's a couple others. We'll just sort of mumble that. But, and we talk about how in general, there's three types and tend to be three subspecies of leishmaniasis in the New World that can cause mucocutaneous disease, mainly Braziliensis, number one, but also Guyanensis and Panamensis. Now, interesting enough, there's a limited geographical distribution. And so on first thought, you would say, well, there's no leishmaniasis hondurensis, so I don't have to worry. But no, unfortunately, there is mucocutaneous leishmaniasis in parts of Honduras as well.
Okay.
So it's really interesting. I mean, I'm trying to figure out how they cursed the city with the mucocutaneous variant of the leishmaniasis, which is really a horrible disease. But what I will say is if you have enough money, IV liposomal amphotericin or miltefosine when I was down in Panama, last week, the way that the local people treat the cutaneous leishmaniasis, theoretically preventing it from spreading to cause a mucocutaneous. I'm not sure how effective this is. The people would put battery acid in the indurated area. Which, if you think about what do, we burn it with lasers, we freeze it, we use thermoablation.
Sulfuric acid, though, isn't it?
We're doing similar stuff to destroy the tissue, as you know.
Or is that hydrochloric acid? I'm not sure which one they use in batteries.
Yeah, I'm not sure. But sulfuric acid. But I don't know. It doesn't need to be as horrible. And you're not going to use that kind of stuff on a mucocutaneous lesion because if it's right at the border, right? Let's say your mouth or your nose or any other sort of area where mucous membrane and skin meet, you're really going to want to use one of these oral or IV therapies.
Yeah.
H2SO4 is battery acid.
That's it. Sulfuric acid. I was right.
Can you take the next one?
I could, yes. Anthony writes, quotes, as first alluded to by Tyner in 1952a, there is good evidence that the parasitic hosts... Paratenic hosts is exactly right. Paratenic hosts, particularly grain-eating rodents, become infected with B. procyon by foraging for undigested seeds and other material present in raccoon feces at latrines. And he gives about 3 different references for that. So their feces, although they can't be infected by it, will transfer the eggs to various other places where people can encounter them. Yes, raccoons can be infected with Beta-Saskers from each other, but that's not the most interesting form of transmission. Rodents are attracted to raccoon latrines as a food source. The larval migrans disable or kill the small animals, enabling raccoons to catch and eat them, so become infected. Here, the parasite is not trying to kill its host, it's trying to kill its host's dinner. Might the arms race paradigm be misguided? I think it's curious, if not comical, if someone tried to explain evolution by looking at a black and white photo of coral reef fish and then using peppered moths as a model. Why is virulence similarly seen as operating along a single axis?
Because it's the easy way out.
Right.
And people are always thinking in their own terms and human terms, and that's what they think about.
Should I continue? Anthony?
Yeah, actually, the next one, he said, forward this to Dickson, and I didn't. So here, you can read it now.
Oh, okay, fine. McLuhan in New York at Fordham University, Friday, October 13th, 2019. And then he gives a reference for that visit. If I remember correctly, you're teaching a photography course at Fordham. Well, actually, no, I'm teaching an ecology course, but I am teaching at Fordham. This event is in Manhattan at the Lincoln Center campus. That's where I teach. So McLuhan's son, Michael, is a photographer. I don't know if he'll be there. In one of the early episodes of your This Week in Parasitology podcast, you mentioned Ulysses by James Joyce. I found this audio of a dramatic reading very good. Wow. I think I'm going to check that out. Joyce's works mentions a virus and quotes letter to the editor and parasites, the fingers blood-stained from picking lice. Joyce was aware of disease, something that McLuhan and Chardin, it seems, overlooked. Burroughs focused on this aspect of modern times / the global village, and he gives a reference for that too by William Burroughs. And one of the beat poets that it was also an author. According to my understanding of Marshall McLuhan, it's not that artists predict the future, but that they see the present, which is a very interesting way of saying that. The illusion of prophecy results when everyone else is looking backwards instead of ahead.
It's an interesting article. Did William Burroughs describe the origin of AIDS in 1959?
Well.
So it's an excerpt from Naked Lunch, and some say the author predicts AIDS. He starts out with lymphogranuloma caused by bacteria, which is associated with AIDS. He refers to a disease from Kenya. He says that the spread is enabled by sexual transmission and worldwide travel. Interesting.
Wow.
Daniel, the next one will be amusing to you. Take that. That's from Veronica.
Veronica writes, thank you, my friend. Still cranking out your podcast. I haven't listened for a while, and I'm happy to hear the familiar sounds of your voices and the intro, outro music. I was soothed. How we relax best with our own most familiar cultural constructions. Last night, I came home from Thanksgiving dinner full of wine, whiskey, turkey, 7 different vegetables, and a piece of cannabis brownie. I'm thinking this is from Colorado. The conversation at the table was very stimulating, thanks to the rather existential state of the world. And my host suggested a little brownie might help me settle down when I went to bed. No, it did not. My mind was racing.
Oh, no.
I searched my fave podcast and realized I hadn't tuned into TWiP for a year. So there was I, high and analyzing your every word and segment change, fluctuation in tone and intake of breath. You guys totally passed the test of close scrutiny. As before, you shifted from the language of biology to humor to empathy and kindness and back to biology. I wrote you before once, and I had to write to you a second time to say hello and bravo.
Oh, that's nice.
I am not a biologist. I got my degree in art and history. I can barely follow your ideas sometimes, but am fascinated by the fact that you all seem to know exactly what you are talking about.
It's delusional on our part.
Somehow I learned something. I didn't have to listen twice to understand that a young New York female contracted a parasite that uses raccoons as its host and burrowed over perhaps 10 years from her gut to her eye. And that, because it wiggled in her eye, the ophthalmologist and his team were able to figure it out, what it was and how it got there. That is amazing. A worm can travel through the human body, transfer through the different membranes and organs without detection, and finally emerge to disturb the eyesight of a teenager. I know schistosomiasis, I think I spelled it wrong, is like that, can penetrate the feet, I think, if I remember Dickson's lectures. I would love to hear a podcast that explains that body thing, how parasites move within their hosts. Love you and thanks, Veronica.
Dickson loves us, Dickson.
That's touching. Nice? That's very touching.
The movement is really interesting, though, isn't it?
Yeah.
I mean, especially for these rather large.
Oh, these are site selection. These are called site selection behaviors, and the body has so many different possibilities that the parasites are selected for living there by their own biology. And that's also, they got two complex biologies interacting to produce a site where the parasite eventually ends up. In this case, of course, the Balis ascaris procyanus doesn't know that it's in the wrong host, so it continues to try to get out. And that's why it's migrating all over the place.
She would love to hear a podcast that explains that body thing.
That body thing.
Can we do
love
that?
To know more about that body thing.
Can we do ourselves. A podcast on that body?
We could do it. We can try.
How parasites move within their host.
I attended a whole meeting once on a day-long symposium on site selection and parasites, just to see what isn't known. Basically, because a lot isn't known, but at least if you can define what you need to know, you can proceed to try to find that out. And everybody presented a little bit different story about their own parasites. And, of course, I was talking about Trichinella at the time. And it's one of the few parasites that actually picks a muscle cell and sticks in it and then stays there, alters the environment, and lives for up to 20 years like that. That's remarkable. And they all came away with the fact that, wow, that's kind of amazing. So much so that they actually put the organism on the cover of the journal that they produced the symposium with. So I was quite honored for that. But
In your video lectures, which are coming out soon, you talk about how all of these parasites enter the host and spread.
They do.
And I think perhaps Veronica would find those interesting.
She would.
For example, how some of them will penetrate the gut tract, then come up and you re-swallow them, or they go in the lung and you swallow them and go into your gut.
That's true.
Or they go in a hair follicle and move. Yeah. So I think that she should listen to those.
Yep.
But they're not out yet, of course.
No, but we're working on them.
And she could watch them as well because they are videos.
We have a rough draft already in the can.
And we have amazing technology where the video is accompanied by talking.
True. So when I say the videos for parasite diseases are in the can, I don't mean that literally, because obviously that's a reference to where your feces ends up.
The film industry, right? In the can.
I know that's their expression, but when parasitologists say that, it means something completely
different. Another TWiP is in the can. That would be a good tagline.
TWiP is in the can.
The TWiP is in the can. Good idea. All right. For reasons that will become clear in the future, I'm going to stop at that email now.
Okay.
Because it is 7 p.m. and we all need to leave.
We do.
I have to go eat my Blue Apron dinner.
Very good. Did you make a Blue Apron today?
It's the chicken sandwich one. I don't know if you're on the same rotation as me.
I'm so hungry. So hungry.
With the roasted carrots and the salad.
You guys.
TWiP, you can find it at Apple Podcasts, microbe.tv/TWiP, or just use your iPhone or your tablet or your smartphone app and subscribe so you get every episode. We'd really appreciate that. If you like what we do, you can help us financially, microbe.tv/contribute, and send us your questions, your case guesses, your
cases,
brownie-induced hallucinations to TWiP at microbe.tv. You can find Daniel Griffin sometimes here at Columbia University Medical School. He's going to be here next week, I believe.
Tuesday, yes.
And other times he's at parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
Dickson Despommier, thelivingriver.org, parasiteswithoutborders.com, Despommier.photoart.com, and so many others. Thank you, Dickson.
Pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. His work is at ronaldjenkins.com. And I'd like to thank the American Society for Microbiology for their support of TWiP, and of course, the sponsor of this episode and the supplier of tonight's dinner to Daniel, Blue Apron. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.